On one side of the operon's three genes are two regulatory regions, a promoter and an operator. The promoter is a site where RNA polymerase can bind to begin transcription. The operator site is where a DNA binding protein known as the Lac repressor can bind to DNA. When the repressor is bound to the operator, RNA polymerase cannot move along the DNA and reach the genes. In other words, the repressor is like a roadblock. The genes that code for the repressor protein, the gene that codes for the repressor protein is located before the promoter region. What's the function of the repressor? It can bind to the operator, blocking RNA polymerase and stopping transcription and therefore stopping gene expression. Locate the Lac genes in the figure. This illustration shows the same segment of DNA in an E. coli bacterium as the previous slide. What's being shown in this picture? The repressor has bound to the operator and is blocking RNA polymerase from transcribing the genes. Find the repressor protein. Is lactose absent or present in this situation? We can tell that lactose is absent, so the repressor is blocking transcription. The repressor binds to the operator, preventing RNA polymerase from binding to the promoter. This illustration shows the same segment of DNA in an E. coli bacterium as the previous two slides. Notice if the repressor protein is always present, how could the Lac genes ever be switched on? The Lac repressor protein has a binding site for lactose. Lactose is present in this illustration. What happens when lactose is present? The lactose binds to the repressor, and this causes a change in the repressor structure. With lactose bound to it, the repressor can no longer bind to the operator. This allows RNA polymerase to bind to the promoter. When lactose is added to the medium, it diffuses into the cell and attaches to the Lac repressor. This changes the shape of the repressor protein in a way that causes it to fall off the operator.